Before we get started with introducing Dean, I wanted to share with everybody brief thing about how you can leverage the Drug Hunter website for your team. Dean is talking about today how to get from analysis of getting from hits to clinical candidates. And one of the hit finding strategies, of course, is a DNA encoded library. And Dean will actually share with us today that only 1% of drugs approximately get discovered through that strategy. So I just wanted to see, you can use the search function here, just navigate to the search tab, and you can use our dynamic search, just start typing DNA encoded library here, for example. You can limit it to molecules, and you'll find molecules that were discovered through a DNA encoded library screen. So let's go into this one here. This is a pan-mutant selective PI3 kinase inhibitor that doesn't show any hyperglycemia. So in those case studies, which is actually the most popular article on the Drug Hunter webpage, what we'll do in the beginning is we'll highlight a little bit why this molecule is important, and what differentiates it from others. This again is a PI3 kinase inhibitor. And with PI3 kinase, typically you have the problem that you generate hyperglycemia when hitting the wild type PI3 kinase, because the wild type PI3 kinase is involved with insulin processing. So basically here is an example of how the problems were solved, how a selectivity problem was solved in this kind of context. So we will show the competitive landscape. You can see other PI3 kinase inhibitors that maybe don't display any selectivity. We will relate the target, in this case PI3K, to the disease that it's being used for, this drug is being used for. And then, as I mentioned, one of the highlights here is we will always share how the compound was discovered, and this compound here was discovered through a DNA encoded library screen, and three things were made sure of. They actually screened the full length construct to make sure that the allosteric network is still in place. They blocked the orthosteric site to make sure that they discover an allosteric inhibitor that could be more selective. And then they prioritized actually compounds that selectively hit the mutant over the wild type to begin with. Then we talk a little bit about how the hit to lead is done and the lead optimizations and what problems were solved during that campaign. As you can see here, we really highlight everything in color. If you want to dig any deeper, you can use any of these purple links here that will link out to the primary sources. If the campaign was structure enabled, we'll show structural data here with you. We'll go deeper in the selectivity. I don't have time to explain this today, but I encourage everybody to read this story here on Drug Hunter. We talk about the preclinical data and clinical data that's associated with the compound. So you can see probably if the preclinical lined up with the clinical, and what kind of other clinical studies are currently going on, where this compound currently is. And in the end, we will always highlight the synthetic pathway of how this compound was made, either in the medicinal chemistry route or the process route. All right. And yeah, we actually find that a lot of people use this webinar here for a lunch and learn. And some people actually also use Drug Hunter case studies for a lunch and learn. Imagine what you could do with those case studies that we have more than 500 of. Rather than doing a quarterly journal club, you could use one of those case studies to discuss at your lunch and learn. And now, without further ado, I want to introduce Dean. Dean, of course, doesn't need actually much introduction. Dean, thank you for being here today. Dean is the Senior Vice President and Head of Discovery Sciences at Jnana Therapeutics, and was previously the Director of Chemistry at AstraZeneca within the HIT Discovery Department, the Discovery Sciences, and the R&D Biopharmaceutical Unit. And he has over 25 years of experience in the industry, both in large pharma companies as well as in biotech organizations. And Dean has been responsible for building many scientific programs in neuroscience, infectious diseases, and rare diseases, and several of which have resulted in the successful transition to clinical candidates. Dean actually got his bachelor's degree at the Abilene Christian University in Texas and obtained his Ph.D. at the University of Minnesota in Minneapolis. Of course, Dean is currently the Perspectives editor with the Journal of Medicinal Chemistry, and I always love Dean's thought-provoking perspectives myself. We actually have a wonderful article about one of them on Drug Hunter. And actually, Dean has also been listed as author and inventor on more than 80 publications and patent applications in medicinal chemistry that include drug design, and that also includes granted patents on clinical candidates. So Dean, welcome to the show. It's really a pleasure to have you with us today.
Thank you very much, Dennis, and the team at Drug Hunter for that kind invitation. It's great to be here, and thanks very much. Look forward to telling a little bit more about some science that I've been thinking about.
Cool. Yeah, no, we're very excited to hear that. Before we get started, we wanted to learn a bit more about you. So
Right.
I was wondering what inspired you to get started in a career in drug discovery?
Yeah, it's a great question. There's a beautiful article that was written in Chem just recently. I apologize, I forgot the author's name, and I think that hits the nail on the head of once I once I started to do a degree in chemistry to realize the power that you had to create molecules that could really shape and help patients' lives and cure diseases, and that really hooked me, and I've been hooked ever since. So I like that power of the idea that a single molecule can really make a difference, and that's really what did it for me. So I actually didn't want to be a chemistry major, but I couldn't avoid it. It was just like my manifest destiny.
Awesome. Yeah. No, that was actually a great perspective. I think Don
Yeah,
Weaver was the author on
that
that
sounds
was. Yeah.
right. Yeah.
Yeah, awesome, awesome. No, I think that really hits the nail on the head. So with that, like what molecule is your favorite molecule and why?
Well, it's a tough one. It's like, which is your favorite kid? Well, I guess the way I'll answer it is that I want to answer with molecules that I've worked on because that's the easy one. I love the current molecule we're working on at Jnana, but
Mhm.
if I look objectively and impartially at things that are out there, I was at AstraZeneca when they discovered osimertinib. I didn't work on it, but it's just a beautiful story of how quick the
Mhm.
discovery effort went, how quick they went through development. It still holds the record of a compound going from IND to NDA less than 3 years.
Wow.
Because they had the right target, the right safety, they just hit the mark, and it was an unmet patient need, and it just shattered the perception of how long it takes. So really beautiful story, beautiful molecule, and patients are living longer lives with it. So I think that always inspires me when I see a molecule like that of like you don't necessarily have to spend 8 or 10 years in the clinic. There are paths that you can get to patients who need it quicker. So I like that one a lot.
Awesome. Yeah, speed in drug discovery becomes a lot more important these days, I think. That's
Right.
great one. So what are you most excited about today in drug discovery? Is it speed or
It's a super question too. I think what inspires me is the idea that small molecule discovery is not dead. In fact, it's alive and well. And that was something that I worried about 15 years ago. And many of us probably in the audience were affected by the perceptions of that. There was some papers written that small molecule discovery was on its way down. It was a Me Too. But if you look, and I'll show some of my slides today, it's far from the truth. Like small molecules are solving rare diseases that we didn't think anybody would think there'd be a market for it or the opportunity. Oncology has just witnessed a rebirth of precision medicine that small molecules are. So for me, it's the outlook of there is a home and a place for small molecules to cure diseases, and it's not going away. So that's what excites me.
I love that answer, Dean. That's also that's what we're preaching to actually
I
at
know.
Drug Hunter.
Yeah.
Awesome. So if you could actually go back to the beginning of your long career,
Yeah.
what would you advise yourself with? Uh,
Yeah. I think it's a motto that I say a lot anymore, and that's drug discovery is a team sport. It really
mhm,
is a team sport, right? And coming out of grad school, you're kind of often a solo artist, right?
mhm, mhm.
And you don't often get how important it is to listen and seek guidance and advice from your peer group. And it took me a while to get that, maybe longer than it should have, but it is absolutely a team sport. Nobody does this on their own. You need a lot of players and help around you. So I think that's the advice that I would give is don't be self-reflective, seek others.
Awesome. Yeah, no, that's also great advice. Also one thing I struggled with too. I think all of
I
us
think
did.
we all do.
Awesome. Yeah, with that, I think we can get started. You can get started
Great.
with the setup of your presentation. And maybe while you're getting set up, the last question that I wanted to ask is, what do you like most about Drug Hunter?
Yeah, great question. I'm going to share my screen and answer that at the same time. So,
Perfect.
as you mentioned, I'm, in full disclosure, I'm an editor at JMed Chem. I love that role. I love the role that JMed Chem and peer review publications continue to and offer to the scientific community and drug discovery at large. But I also love what Drug Hunter does is provide this outlet, this forum where other content that might not ordinarily be packaged together in a peer-reviewed journal can be seen, can be viewed, interacted with. Some of the content you guys have done to put some of the tools that medicinal chemists need. I still have the mouse pad on my table that has all the different kidney filtration rates for different species, and so I think these two are complementary, and that's a great place to be. That there's a place for peer review, and there's a place for content that medicinal chemists can go to and go to quickly and publish quickly and seek, and I really like that.
Awesome. Yeah, thank you so much, Dean. And with that, yeah, if you want to put it in presentation mode,
So
without further adu, ladies and gentlemen, Dean Brown.
thank you very much, Dennis, and the folks at Drug Hunter. And I'm gonna hide my self view here so I don't have to look at myself and close this. So I'm not sure I'll be able to see in presentation mode comments that are coming up, and I will leave some time for questions. But I'm really happy to give this talk, and just as a prelude to this, and going back and thinking about, one of the questions that Dennis asked of what did you learn early in your career? Early in my career, I was really blessed to be surrounded by some amazing mentors and folks that would just give me unsolicited advice. And I kind of miss that, right? That was a great part of growing up in the industry. But as I started to grow up in the industry too, I started to think about some of the stuff that was said to me and wondered if it was just like conventional wisdom or if there was actually some truth to it, or because of the battle scars that people had endured, if what they were telling me was real and true. And I think that's a healthy thing. And so I got on this crusade a few years ago to start studying some of these things that I've been told through my career, and just see if there's something behind it. And so with that, I'll jump into the first slide. Oops, my slides are not advancing. Oh, okay, now they are. I'll be real careful about clicking. And so with that backdrop, as an example, some of the things that I've been told in my career are shown in the first top bullet points. Maybe you have been as well. And one is that quite frequently the development candidate is discovered early in the project, and you spend many years and many compounds trying to find a better one. But another part of that, and this has been told to me before, is that your development candidate is usually pretty close to your hit structure, right? And so, as a spoiler alert, I don't have an answer to this first bullet point. I hope you can see my mouse. If anybody's interested in writing that article, I'd love to collaborate, because I think that article would be a collaboration with different folks in the industry talking about how long it took them to get to their clinical candidates. I don't have an answer for that, but I did dive into this question of what do the clinical candidates and how do they resemble the hits? And as you start digging into that information, there's a lot of other content that you can exhume, and I'll show you that as we go. So there are two papers that this comes from. One was a collaboration with a former colleague, Jonas Bostrom, where we looked at 60 publications from JMed Chem, and the more recent one was 156 examples from 2018 to 2021. And as I go forward, I'm going to put some caveats on here just so you understand the approach that I took. And there are limitations to the way that I did this, and I'll be fully transparent around that. But I still think there's content and education to be taken here. So as an outline, we'll talk about all of these case studies and look at the lead generation strategies that were employed. We'll examine the target classes and disease area distribution, spend time on what changes in properties have occurred, specifically log P and molecular weight, and then getting into similarity and trying to answer that question, like, is it fairly common for the clinical candidate to look very, very similar to the hit, or is that an exception? I'll speak briefly about rule of five, and how some of these molecules adhere or don't adhere to it. A couple of case studies, and then along the way, I sprinkled in some hopefully provocative slides that will get some discussion going as we go through this. So just to start out and to put some caveats, how did I go about this? I did use JMed Chem as the source here. I could have used other sources, but in full transparency, it was just a lot easier to use a single source. There's lots of other journals out there that have case studies, and I thought it would be a representative slice. So I stuck with JMed Chem for this analysis. I suspect if you did it with other journals, you get the same download. When Jonas and I first did this, we started to try to figure out how do you categorize stuff? Like what is a hit finding method anyways? And it does become somewhat subjective, right? Depending on how the author called it. And so we came up with this list, and I refined it later with the subsequent publication. So one big category is known, and we'll talk about that in detail in a moment here. The other one would be random screening, and I think we all know what that is, but I didn't delineate whether it was 200 compound random screen or 2 million compound random screen. I just used that as the category. Directed screen would be something like you're screening a kinase and you buy or you have a kinase directed library. Fragment screen, virtual screen, and DEL. I think we don't need to explain what those are. And in some cases, multiple hit finding strategies were used. And with those, I just captured them all just to get a running table of these are the hit finding approaches that were used. So the caveat that I'm going to put on this is not a referendum on any of these approaches. As you'll see, some of these approaches didn't have very many examples, and there may be reasons for that. And I will actually go to the next slide and explain that in more detail. So this is the initial tranche of all of those hit to lead programs. And it's consistent not only with the previous publication that Jonas and I did a few years back, but it's also consistent with, whoops, I misspelled Genentech. I really apologize about that. So it's a similar Roche Genentech analysis that was published that the vast majority of clinical candidates come from known starting points. And we'll go into detail of what that means. 21% come from random screens, and then the rest are smaller subsets. And this is where I want to point out, it's not a referendum. I used to do a lot of DEL screening at AstraZeneca. I'm a big believer in it. It works, but there just aren't that many case studies of clinical candidates yet, and there could be reasons for that. Either the technology is a little too early to deliver a pipeline of clinical candidates consistently. It is a proprietary technology. It may not get published as much. It might be that those case studies actually go to higher impact journals and they're not in JMed Chem. So there may be reasons why that's the case, but I don't want anybody walking away from this and saying these kinds of strategies don't deliver. That's not the intention of this article. They can, they will, but this was more around what are the more common strategies. So in the previous paper, I also looked at the use of structure-based drug design, and Jonas and I had that as a separate category, or had that as a category, but it made more sense this time just to pull it out on its own, because as you can see, most projects actually do use some type of structure-based drug design, whether it's a bona fide crystal structure, they're ligand with the protein of interest, or if it's a homology model, or even if they're using other types of structure-based approaches, 65% of them report using that and 35% don't report. So it's possible they did, but they didn't report it. My take-home message from this, and based upon what I've seen in the past, and this is stating the obvious, is that structure-based drug design is probably going to increase with the improvement of cryo-EM and the ability to get structures on membrane-bound proteins. I think it's only going to increase as a capability, as a tool finding capability. But these are the numbers, and we're going to dive into that in a little bit more detail. So what does it mean to come from a known starting point? Well, I guess before we do that, let me just show you how we've cataloged these examples just to orient you. So if you look at the left, here's an example that was a category of a known hit. This is a selective estrogen receptor degrader from AstraZeneca. The hit was actually an AstraZeneca compound, I believe, that didn't have the right properties to make it all the way as a drug. And so the team reimagined it and reinvented it and replaced the cinnamic acid portion with a basic amine and a floral tail and came up with AZD9833. So that's an example of a known starting point came from the sponsor itself. Here's another example, and I think it was cool to put these side by side. Same target. This came from a random screen, right? And so this is, you could imagine the diphenol here mimics the estrogen itself, and it's not too hard to imagine that that would be a hit. They ended up at the top part of this at a very similar place as AZ, but their core structure was vastly different. This is a Sanofi example, and this came from a random screen. So just to orient you on how we classify a hit, we track down the hit. It led to that development candidate from that literature. Here are a few more examples, just two more. Here's a random screen. We're going to talk about this one in more detail. This is a quinoline aldehyde that led to the FGFR inhibitor roblitinib, a really, really elegant example. And we'll talk about why that aldehyde was so critical for oncology. That came from a random screen. Here's an example from Agios that highlights fragment-based lead generation, so a hit that ultimately became the clinical candidate from Agios for MAT2A. So that's how we cataloged all of that information. We found the hit, found the candidate, and then did a physical property analysis and a similarity comparison between the two discrete molecules. So it's worth spending some time on what it actually means or how we define what known is, because I think it also teaches us an important lesson in the industry. So when we put all of these into that bucket, it turns out there's at least 4 or 5 flavors of known. And I think the immediate thing we think of is it's a patent busting exercise. And actually, there are very few of those, and probably not surprising. Like, nobody's going to publish a patent busting exercise in a peer reviewed journal and open themselves up to IP issues. And perhaps they're not as common as we think they are too, but there's just not that many examples in the literature. What there are a lot of examples of are these, where there was an existing clinical candidate where they observed, or a development candidate, maybe it didn't get in the clinic, where they observed an issue and that informed them to make a better compound. So here's an example from AstraZeneca, LAP inhibitor. This was predicted to have a very high human to dose projection and clearance in cardiovascular issues. This actually, I believe, came from a virtual screen way back when. If you keep tracing it back, I believe that some of the hits here came from virtual screening. But this was the lead structure that was very advanced, and they were able to tackle that with reducing hydrophobicity and getting a better molecule that gave a better human dose projection. Other examples, this is one of my favorite, not my favorite, I'll show you my favorite in a minute, example of a clinical candidate that really informs. This is the Peloton HIF2 alpha inhibitor that they later licensed to Merck and is on the market. This one's belzutifan's on the market. But when this went into clinical trials, they realized that the human dose predictions were wildly off, and that there was an enzyme that glucuronidated at UGT2D17 that really decreased the actual plasma exposure and limited therapeutic efficacy. So it's amazing what a fluorine atom can do. Just moving that fluorine over and making that particular diastereomer eradicated UGT, but they didn't see this preclinically, right? So they had to go into the clinic to understand this. And I think that's a common theme and an important consideration to those in the field, that quite often you need an initial clinical candidate to test your hypothesis. Here's a GSK example in S5B for HCV. They ended up with a metabolic soft spot that limited the human clearance that was insufficient to go forward. So they redesigned and focused on improving the metabolic clearance that led to GSK 175 with a much improved human half-life than the original. This is actually my favorite example, really cool story. We encourage you to read it. It's a Novartis compound for leishmaniasis. And this is a third world neglected tropical disease. So it has to be very economically viable. This was the initial clinical candidate that went forward, but it turns out it's very insoluble. And if this were for a first world indication, for cardiovascular, for diseases in other parts of the world, that would go forward, but it would require a very expensive formulation. And that's just not practical for these types of neglected tropical diseases. They have to be distributed to poor rural parts of the world. And so they informed themselves in development that what they needed was something that was kinetically more soluble. And so just the simple orthosubstitution, many of us have learned that trick, led to this LXE408, which did go forward. But they didn't have that information in hand until they developed that compound and moved it forward and realized that it just was a non-starter for that particular economic situation. So known compounds come from, a lot of them come from existing experiments in clinical trials. There are more conventional approaches, and there's a few here of when we think about known that come from the literature. Here's a Gilead example, TLR8 agonist that came from some literature starting points. And this Pfizer molecule, I think this became part of Cerevel, if I'm not mistaken. They claim that this molecule was inspired by pharmacophoric elements on GABA modulators in the literature. They designed this compound, and off of that, they designed in the potent and selective GABA-A compound that went forward into clinical development. So known starting points also come from literature. So it is important for those that publish in the field that hit dead ends when they publish. Sometimes somebody else can take that and move it forward and do what's best in the best interest of health and society. So it's quite important to publish results if the sponsor can no longer move them forward. So shifting gears a bit, and we'll come back to some of those topics in a moment. Extracting all this data, you also get to look at target class and disease area distribution, and there probably are no surprises to the audience here of the types of target classes that are represented in these pairs. Kinases make up the vast majority of those. I chose a category called other enzymes. There's about a dozen or so in here. So if you read the paper, you can see the actual itemized enzymes, but combined, more than 50% of those descriptions of the case stories, and in some respects, it's a leading indicator of what's coming to the market, are addressing enzymes and kinases. GPCRs are a distant third, and that was different 15 years ago, 20 years ago. GPCRs, ion channels, and transporters were much more prominent, became much more part of the field. So you can use these analyses to see also where the field is moving. And as we look at disease area distribution, again, no surprise to many of you that oncology tops the list. Might be a surprise that CNS pain is second in the list as well. So that's interesting to me and surprised me because the field kind of moved out of CNS pain many years ago. Large pharma began to make some strategic decisions to limit their exposure there. What's happened is biotech has kind of replaced that to some extent, both in drugs coming to the market and in terms of the number of programs that are being worked on. And so by and large, CNS pain still commands a pretty sizable portion in disease area distribution. Immune and inflammation is also leading behind, and it's ticking up every year if you look at sort of the number of publications and the number of compounds going in the clinic. I'll just say one more comment on this slide, and that's infection. If you looked at this 10 years ago, infection was second in terms of drugs and things that are being worked on in clinical candidates. Some of those have been solved. HCV has been a good example where there isn't a lot of active work there, but there certainly was, and HIV certainly was many years ago. So this gives some leading indication of the disease areas that are being worked on. So we're going to shift now into the physical properties because this analysis allows us to look at the changes in hit to clinical pair for the examples that were pulled out. So I had to restrict this to the largest categories because in fragment screening and directed screening and cell screening, there were so few examples that it didn't really, I think, portray the changes as much. So I restricted it to known and random. And I also think that's the most distinct pair to look at and the most interesting pair. So this is the change in C log P for any given hit to clinical pair. And it's broken down by known in the green, okay, and random in the red. And so when I put this data together, the thing that jumped out to me is, if you look at the averages, there's not much change. The average tends to be around 0 change in C log P, but that doesn't tell the whole story, right? There are many examples on the right-hand side of this histogram where you're adding lipophilicity to the hit to get your clinical, but the thing that jumps out to me is there are very few examples on this side, and even fewer the more lipophilic you get, of starting with a hit structure and lopping off a substantial amount of lipophilicity, right? This bar graph is weighted more heavily to the right-hand side. I'll show you one example. There are not many. Here's an example in the PARP field where this is from AstraZeneca. This is the random screening, the starting point, a C log P of 4.06. This was the ultimate the final clinical candidate, and that was a delta log P of minus 2.32. So it can happen. I don't want to say it can't, but the examples are rare. And I think we all know the difficulties and the challenges of starting with a high lipophilic point and trying to move away from it. And I want to build this case a little bit more and show you some, at least one slide of unpublished work that we did at AstraZeneca. And this also exemplifies many other HTS screens. So I worked very intimately with our HTS screens at AstraZeneca, and we did a lot of them, of course. And this is one example, but most are like this. And this was a norepinephrine reuptake HTS screen that we did many, many years ago. And if you look at the random hit rate, the hit rate at large is about 1.25%. So if you screen 2 million compounds, you're going to get a hit rate of about 1.25%. But if you then bin it and say, how many compounds did I have in my collection that had a C log P of 5 to 5.5? What is their hit rate? It's that. Well, what if I bin the compounds in my collection that have C log P of 1 to 1.5? It's down here. And so you can theoretically argue maybe that's a better starting point, because probably I'm going to have to add lipophilicity onto it. But as it turns out, I get a 12 times higher hit rate for compounds in my collection that are that hydrophobic. And so it makes it difficult if you don't have many of these compounds in your collection. That puts you in a disadvantage. But a further disadvantage is if you don't want to start with this kind of stuff up here, chances are you're going to have a higher hit rate and you're going to find those things as starting points. So I think the quality of the library is critical. And I hope I'm going to show a joke on the next slide. I hope it translates across the globe. I realize we have a lot of folks from different places, but this maybe will help stick it in your mind. Don't take it from me. Take it from my heroes, John Travolta and Olivia Newton-John, that grease is the word. And hydrophobicity is this double-edged sword, right? It's a great fishing hook. If your library has hydrophobic compounds in there, they'll probably find hits. But it's hard sometimes to get away from that. It's hard to cure that addiction. And I hope I convinced you of that, showing you on the hit to clinical histogram that it is hard to get away from it. It's not impossible, and people do it, but it is hard to get away from it. I see a smiley face emoji. Thanks. At least someone laughed. Excellent. So the next slide is molecular weight changes. And this one has some similar trends to the C log P. And I categorized it between known and random, again, to take advantage of the largest buckets of information that we had. And what you see here is kind of what you would expect. I think if most medicinal chemists guessed at this, this is what you would expect to see, that the known starting points shown in green in this example, on average, added about 30 molecular weight units onto that. And it makes sense, right? So if you're starting from something that's heavily optimized, lead optimization program, you're fine-tuning it, you're tweaking it. You might be adding a fluorine, you might be adding a nitrogen. There are some examples where you're adding a lot more and some where you're taking away, but on average, you're adding about 30. With a random screening collection, you're on average adding 80 molecular weight units, but it's also more broad and distributed. There's as many examples at 80 as there are at 120 to 150. So I think, these two things in my mind sort of connect together, that lipophilicity, molecular weight, and this is stuff we all know. So, I hope I'm not overstating the obvious, but the quality of your library, knowing that you're going to have to add on those elements, most likely, becomes critically important. And again, apologies for overstating the obvious to many of you. This is the analysis of similarity, and the way we did this is to use the Tanimoto maximum common substructure. There's some tools out there you can use to do this, but basically what it does is find the common pharmacophore between the two compounds that is the most similar. So a 0.9 would be something that's very, very similar. So for example, it would be adding a fluorine, perhaps, or one nitrogen atom to the core to make something that close in similarity. Whereas if you go over here, the Tanimoto coefficient is much smaller. These are less similar. And so again, I categorize that by random and known. And I don't think, as you squint at this graph, that there's a whole lot to take away in terms of these are substantial differences between random and known. The takeaway for me is there's a whole lot of diversity from what a hit clinical candidate leads to a development candidate. There are some, even on the random side, that are really, really close, and we'll show you those examples here in a moment. And some are more distant, and it's what we do as medicinal chemists, right? It is our job to take the journey and figure out how to get from point A to point B. And I'll show you some of the examples. What about incorporation of other atoms? This is the same sort of analysis, right? That here I didn't distinguish it between known and random. I just bunched it all together. We have hits here in red. So 30% of the hits have a fluorine atom on there. 60-some% of clinical candidates have a fluorine atom. So again, that's hopefully not stating the obvious. Many of us do this. There's some great reasons to incorporate fluorine into your hit. A lot of people do that. They don't do it so much with chlorine or sulfur, and bromine is very underrepresented. And I think there's obvious reasons why these are not added to your molecule in excess from your starting point. But it also begs the question, like, is it best? What if you had a library that had, a lot of fluorinated analogs? Is it best to have that, or is it best to have a library that doesn't, knowing that medicinal chemists will pull those in and put them where needed? It begs the question, and I don't have an answer for that, but I think it's an interesting, provocative question. So that's the incorporation of atoms in terms of distribution. Briefly, I'll talk about rule of five, and just in full candor, I'm not a rule follower. I don't believe in rules per se, but there is something here, right? And I don't want to ignore it, that most clinical candidates, and it's true for most marketed drugs, they do follow the rule of five. 79 out of 138 have 0 violations. 43 have 1 violation. These are mostly molecular weight and hydrogen bond acceptor. Very few have multiple violations. So it's not impossible, right? There is a whole field of beyond rule of five, and it can be done. But the probability is higher, I think, if you have fewer of these kind of potential liabilities in your molecule as oral candidates. So just bear that in mind. I think that it's useful information to not be overused, but it does hold up. And this is a paper that we published a few years ago on marketed drugs, and it's the same distribution, and it hasn't changed over the years. So again, don't interpret this as it can't be done. It can be done. There are great examples of beyond rule of five drugs that are out there, but most of them do. So if you're given a choice, I think I would choose one that didn't have liabilities that might preclude oral absorption or the like. The other trend that is very observant is covalent warheads. So about 10% of the hit to clinical candidates contain a covalent warhead. Most are Michael acceptors, but there's oxyborols, there's some epoxides, there's an aldehyde that we mentioned. We'll talk about it in a little bit more detail. I saw a really cool, I think it was an aziridine from Revolution Medicine on the AACR get shown on social media. So covalent warheads are picking up, and some of the hit to clinical case studies started with a non-covalent and built in the covalent. This is evobrutinib, and this is the BMS branibrutinib that followed that path to get to the covalent warhead. But there's some really awesome examples of doing it differently. So this is from Mirati, KRAS G12C, and this is the Amgen compound, same KRAS G12C. I'm sure these are both profiled on Drug Hunter. And they used a covalent fragment screening approach as part of their integrated drug discovery capabilities, and landed on some very, very sophisticated and complicated molecules, and came about it in a bit of a different direction than the previous one. So covalent warheads, covalent chemistries, definitely on the uptick, certainly more so than it was years ago. So I'm gonna kind of speed it up just a little bit, but show you some examples of what we mean by ones that have high similarity based on the Tanimoto distance. These are all random examples, random screening examples. I didn't think it was fair necessarily to do this on the known examples. So here's an example from Galapagos, this ADAM TS5 inhibitor. As you can see, the clinical candidate is a spirocycle incorporation at the alpha position and a methylation and 2 fluorines. That may seem trivial. My guess is it took them quite a while to get there. There was also some published literature that may have helped inform that as well. But as you can see here, here's an example where the hit and the clinical candidate aren't that far apart. So it was a high quality hit. Another example, EP4R for endometriosis, a Bayer compound, as you can see here. They got away from the methyl substituent, the methylamide, ended up with carboxylic acid, and have a different substituent off of the benzimidazole nitrogen and a methyl substituent here. So not a substantial amount of difference. The pharmacophore is largely intact. The overall C log P is 0. Bear in mind, I didn't calculate the log D. That's obviously acidic, so that will change that. But this is not very different as reflected by the Tanimoto differences. One more example, lanifibranor, which is a pan-PPAR activator. This one incorporates one additional methylene unit here and a heterocycle alpha, the sulfonamide shown there, which gives you a very close similarity to the hit candidate, and that became the clinical candidate. So there's 3 examples where what came out of your collection was 1 or 2 or 3 modifications away from your clinical candidate. Here are some that are not. So these are examples where that Tanimoto maximum common substructure is much smaller. This is that aldehyde we talked about, roblitinib, quinolinaldehyde, and it turns out, and I'll show you this in a minute, when you do the Tanimoto analysis, it actually picks up on this piece here, okay? But that's not the critical piece. The critical piece is the pharmacophore, and it became very important. It engages a cysteine in that pocket. It's very selective. They can't replace this with traditional Michael acceptors. That aldehyde becomes quite critical. So that's another learning point, is sometimes you pick up a pharmacophore that you can't lose throughout it. It's like the hook throughout the drug discovery expedition. And in this case, it was the aldehyde for that team. Here's a different example, very low Tanimoto maximum common substructure core. This is the hit, and you look at the lead. I'll show you that in a little bit more detail. There's really nothing in common between the hit and the clinical candidate. And a really nice example of what medicinal chemistry can do, just inspired by a single starting point. And an elegant example from Novartis SHP2 inhibitor. This one may be a little bit more intuitive as a medicinal chemist. You can think of this different pharmacophores here that you might modify, and they change the geometrical isomers of that pyrimidine, but also added very different substituents on the left and right-hand side, guided by structure-based drug design. So here's an example where, again, it's that pharmacophore, that key pharmacophoric element that really drove the drug discovery effort. And just kind of to summarize those points, here's that roblitinib story. This is, as I understand from the literature, I haven't spoken with the authors here, I'd love to at some point, this seemed to be the key, that quinolinaldehyde, and everything else was more optimizable, but they knew that was critical for activity. So it's this key pharmacophoric element that they sort of anchored on and did the drug discovery around that. Here's the example from Bayer that I mentioned as well. And this is the actual maximum common substructure. It's just basically a phenethyl acid that they eventually changed at the end and replaced with this methyl trifluorosubstituent here in a chlorophenyl. And I think they did this without structure-based drug design. So it does highlight what you can do from a starting point to the finish in the absence of a good crystal structure. Here's the Novartis example. I want to just spend a few more minutes, maybe leave you with some provocative thoughts. So when I think about drug discovery, to me, it's sort of prospecting for gold. And I like this slide because it sort of illustrates, like, if you do a screen and you end up with random screens, these might be your starting points, but you have no idea how deep the valley is. You have no idea how deep the ravine is and where the gold is. So you're just going to kind of pick away at each level, and then you have to make a call, and you have to make a decision of, should I go any further or should I back away? And so it does create the idea of, do I just scatter this with a shotgun, or do I take one of these and go deep on it? But it also really stresses the importance of your library. And that was one of the things that I highlighted previously, that the quality of your library will dictate what you get. And that's an obvious saying, but it really is. But also the tools we use become important. It's an article we wrote almost a decade ago, but it looked at the medicinal chemistry tools that we use and the change over the past few decades. These are, the production reactions. That's the last reaction you use to make a compound that goes for testing. A lot of amide bond formation, Suzuki, SNAR2, Bach deprotection. This is kind of where the industry is going. But if you look in 1984, it was heterocycle synthesis. Now this is a little bit overplayed, right? We can buy a lot of these heterocycles now, but it was a very different style and different way of doing chemistry. Because of that slide, I have to show this next one, and that's, if you've never read this paper about Leo Sternbach, you should. Great reflection of a tremendous influencer in the field. But I love his statement that discovery of benzodiazepines was done on the chemically most attractive, challenging, and satisfying way. And that's how he landed on the benzodiazepines that made such a difference in patient lives. But it's different, right? It's a different drug discovery model. Right? It was driven by in vivo models then. Now we have high throughput in vitro screens, and there is a demand to make more and more compounds. And also, as we go forward, do we have the right tools in place? And it's also a reflection of those of us that have maybe come from a natural product background. The tools you use there are very different from the tools you're using now. If you look at natural product synthesis, a lot of silyl D protections, oxidation of alcohols, aldol reactions, those just don't get used much in med chem reactions, certainly not in the last step. We favor towards amide bond formations. Bach bead protections and hydrolysis and things like that are common for both. But I can't remember ever using a Grignard reaction sort of in the final step to make a molecule and make a library of molecules. But if we did, that might be a really interesting library, more hydrophilic, more diverse. So just some thoughts on how we take this information and package it together, that there are things that we can learn from the literature. What tends to happen, and maybe I'll pull this all together here on the last couple of slides, that if you're thinking about making a library, and this is just a very generic library where you might use those reactions, the Suzuki and the amide reactions, it's actually quite hard to make molecules that hit this level of log P if you want those, right? And remember the distribution that we showed on some previous slides, that the hit rate is substantially even lower for that. So you have fewer of them. The hit rate is lower, so it's no surprise you don't discover those. They're hard to make. The lipophilic substrates work better. So I do think there is a call here, I think, in the field to really kind of reimagine how we're making libraries, how we're doing chemistry. Is it the right way? Are we doing the right thing? And as an industry, we've sort of defaulted to these large libraries, a more is better approach, high throughput assays, this demand for fast turnaround time. And I know a lot of you have felt this, and we have a heavy reliance on few reactions. And then, this sort of take home message that a lot of clinical candidates come from known starting points. And I don't want to leave you on a negative, because that's sort of rounding up to a negative, but it's not. And the question is, how does it work for us? Well, it actually works. So if you look at drugs that are coming to the market, small molecule discovery is alive. It's working. That model that we put together is working. Will it continue to work? I don't know. Is it the optimal model? I don't know. Can we do better? Maybe. I think we might be able to, as we think about the next wave of targets, the hard to drug targets that come along. So the model works for us, I think. I hope you accept my opinion on that. And not just the drugs coming to the market. These are a distribution of first-in-class versus non-first-in-class medications. And over the past few years, it's 50-50. So the drugs that are coming to the market are novel. They're for new indications. They're challenging targets. So the model we put together works, but I do want to leave you with the challenge of, is it going to work in the future? And are there ways we can do it better? What about all the targets that people have hit their head against and can't progress? Is there a path forward? So I'm going to stop on this slide and just summarize that the hit the clinical candidate approach gives an opportunity to kind of reflect on in the industry and what's working, what we could do better. But as we tie that together, those are drugs that are coming to the market. We do reflect on the fact that it is working, but will it work in the future? I think is the key message for everyone here. So I'm gonna stop there and realize that I went over time just a little bit, and I guess we have time for questions. So thank you.
Awesome, Dean. Thank you so much. That was a really phenomenal talk, and I'm really glad to hear that one of your favorite examples was the LXE compound that was discovered at GNF, actually.
Okay, great.
I worked on it a little bit to
Oh,
optimize later. Thank you.
Cool.
So
we didn't plan that.
Yeah, we didn't plan that, of course.
Yeah.
And I really like the aldehyde story too, that you shared. I think voxelotor is another great example for a reversible covalent aldehyde that Pfizer now bought from Global Blood Therapeutics, I guess. So thank you, Dean. And I guess let's go into the Q&A box. For everybody, please put your questions in the Q&A box if you have them. I see a few questions were actually coming through in the chat, but we're going to start in the Q&A box. So the first question comes from Gil, and he's wondering, when you analyzed the rule of 5 violations, did you filter for orally dosed development candidates only?
I did. I thought that was the fair way to do it. And sometimes it's not so easy to pull that out. Like the authors in some of those publications, they don't always say whether it's going to be an oral indication, believe it or not, because they may not know at the time going into the clinic. Certainly by the time they get there, they know. But I did specifically filter for those that explicitly said an oral.
Yeah, that makes sense. Next question comes from Ann Tran from Amgen. And she's wondering, when comparing the hits and clinical candidate pairs, especially the ones in which hits and clinical candidates have significant difference, do you observe a general increase in fraction SP3, or whether is it
Yeah,
dependent on disease area or indication?
that's such a great question, and I know I know that's a popular topic. We did it in our first paper, Jonas and I, but it was a smaller section, and we didn't see much difference going from fraction SP3. So I actually did not do it on this last analysis. And just if you visually inspect and look at the molecules, there's not too many that do. So it may be another opportunity as medicinal chemists recognizing the world is 3D. That we do sometimes not realize that, but it wasn't evident in the statistics. Great question
Cool.
though.
All right, we have a few more questions. Anonymous attendee is asking, so let's see how the question is. In starting, it was told that hit to clinical candidate is not much change. However, in examples, it is said we start with hydrophobicity and we need to move towards balance. Oh, I guess that it was not really a question. Sorry. I guess more of a comment.
Okay.
Another question from Chris Brown is, did you exclude prodrugs in your analysis?
Yeah, a great question from Chris. I have them in there, but I excluded them out in the analysis. So I included them in the paper if you're interested. They're part of the tabulation, but I didn't think it was fair to include them in the property changes that I was examining. But there are quite a few of them, and that's interesting, right? Prodrugs are definitely have a spot in drug discovery. Sometimes you get talked out of them and said don't do it, but people do it. They get successful. There are some examples there, but we didn't do the analysis on the changes.
Yeah, no, makes sense. We recently highlighted one that selectively goes into the brain. It's like an acid product. It's an ester that
Oh, wow.
goes into the brain and then actually sticks in the brain. So it actually has more brain exposure there. So kind of
Oh,
a really
wow.
fascinating compound from Nottera. It was an NLRP3 inhibitor, I
Wow,
believe.
that's really cool. That's a really cool example. Yeah.
Yeah. So let's see. Another question from Xiun Chen. Some PROTACs break the rule of 5, but
They
they have
do.
good PK properties. Is there any guidance for those?
Yeah, it's not my it's not my area of expertise, but there certainly are examples of doing it. And I do want to go back to, I hope nobody interpreted it as me saying you cannot break the rule of 5. You can't. If there are examples, PROTACs are, but in this analysis there weren't any PROTAC examples. I think the field was too early to publish these kinds of examples. There are only a handful in clinical trials right now. But it will change, and I think it'll change that analysis. And you'll have to talk to a PROTAC expert. I think there are ways of shielding some of the hydrogen bond donors and things of that nature, but or hijacking a transporter. But it's a big field that's emerging. So good question.
Yeah. No, definitely. We're seeing a lot more macrocycles and those types of molecules
Yeah.
moving into the clinic.
Yep.
PROTACs, of course. Hopefully, we'll get the first approved PROTAC this year. That would be very exciting.
Yeah.
I guess along those same lines, I'm going to just read a question from the chat. Could you please comment on the rule of 5 adherence and beyond rule of 5 compounds as they relate to current chemical space and where chemical space is heading in order to address new previously inaccessible targets? So also like a very challenging question, I
Big,
guess.
big question, but one that I'm highly interested in, and I just it's hard to nail it down, right? How do you define chemical space? And we previously used these little triangle plots, the PMI plots, and it's okay, but it's still a little bit superficial, in terms of what chemical space looks like. But chemical space is like 10 to the 60th, like it's enormous. You think about what that is. It's like what we make in a screening library
Hmm.
at any big company is like the size of a postage stamp compared to the US, right? That you're making a couple million compounds compared to 10 to the 64th. So I've always kind of been daunted by that challenge of how, how does it actually work? Like, how do we do what we do given given the
Yeah.
odds, right? It's pretty amazing. And Dell's the same way, right? It's amazing. You can make 20 compounds out of 2 billion and get there, but it does work. But I do think some of the things I was alluding to in the end are really important points of thinking about more diverse libraries and using different chemistry to get there, and specifically around more hydrophilic space because it's really easy to make hydrophobic compounds. It's not so easy to make the hydrophilic ones, and our libraries aren't populated with them. So the simple thing could be just populating our libraries with more hydrophilic. It's easier said than done, though. So
Hmm.
that's I think the simple answer to it.
Yeah, yeah, that makes a lot of sense. Then staying on the topic of high throughput screenings, do you have any types of assays as personal favorites in terms of predictive power? For example, using purified proteins versus cell
Mm.
lysate, intact cells, or phenotypic screens?
Yeah, great question. I think the best answer for that is on any given project, you want the assay that is the most close to the biology you're studying, right? And that's easier said than done also, right? Because sometimes you can't develop that assay in high throughput. So you have to develop an assay that's more high throughput, but gets further and further away from the biology. And that's where you can run into trouble if you're not careful because you may be picking up artifacts, you may not be reading out the biology. So as a blanket answer to that question, my favorite assay is the one that most closely mimics and represents the human biology. Not always possible to
Yeah.
do, but sometimes you would favor, I think, using those assays lower throughput than a high throughput assay that's some, Frankenstein's monster of different things that is maybe more of a detection assay that you might use on a high throughput screen, but you might not want to use to drive medicinal chemistry. So I hope that's useful.
Yeah, no, I would 100% agree with that. That was also always kind of my philosophy as well. So a lot of people are wondering about AI and machine learning and drug discovery. So few questions we hear, and I'll just summarize them. Is what is your opinion on basically using machine learning and AI-based drug design? And there have been recently a few cases, but
Yeah.
it's not making it into the SAR, and like how would you see that for the future of drug discovery, drug design? Where could AI and machine learning play a role there, and how could that influence maybe
Yeah.
the analysis that you did?
That's a great question, and I've wrestled with this, and we all do. Um, it's I'm not an expert in the field, so please take everything I say with a grain of salt. But to me, it's a tool in the toolbox. You'd be remiss if you didn't consider applications of it. But on the other hand, I think assuming right now that AI will discover a drug is probably not there. I haven't seen case studies that really convince me of that, but there are great examples for very discrete problems. I think AI, for example, in predicting, if you were predicting soluble, something very discrete example, are machine learning methods to do that. Those have been proven. Machine learning methods have been proven to do that. But coupling that with AI that would then suggest like the next molecule to make that's not insoluble makes a lot of sense. And I think it's doable. The challenge, of course, is drug discovery is a multi-parameter optimization. So if you focus in on one discrete thing, you may be designing in hERG, you may be designing in other things. So I think that's the challenge for those tools is to get to where it can not only handle like one discrete problem, but multiple problems. And again, no expert in the field, but I don't think we're there yet.
Hmm.
Hopefully we will be in the future, but because it only benefit patients, when we get there, and that's what we're doing. That's what we're all about. But I don't think we're quite there yet.
No, that makes sense. I think I agree with that statement. I think now we're on the top of the hour, Dean. How do you feel? One more question, maybe, or
Yeah,
you want
sure,
to
that'd
wrap
be
it up
great.
here? Yeah.
I'll
Okay.
take, I'll take.
Let's do one more question and then we'll wrap it up, everybody. The last question then would be, what are your thoughts on the high throughput library or screening collections of large pharma companies compared to like CROs and smaller companies? Also a
Yeah,
great question.
such a great question. I worked in both, and they both work. Like the bottom line, they both work. Like you can buy high quality CRO collections that will find you hit candidates. They're out there. They do work. Um, Compound collections and big pharma work, and there are more of them. Like they'll have millions of compounds, but they do get sometimes bogged down by like how many they're supposed to screen, because screening 2 million compounds can take a long time. We can get a lot of noise, so oftentimes they'll even pare it down to smaller subsets. But I think most pharmaceutical companies have gone through over the past decade or more just recreating their collection, because the collections that were in the early 2000s were wrong. Combinatorial chemistry, we went through that at AZ, but when I left AZ, it was the collection that we built was delivering. It worked really well, and I can say the same for biotech. So I think you can get them both, from both sides, but I do think there's more to learn, right? And as I mentioned, this idea of libraries that address some of the critical needs, those are not out there as far as I can tell, and I think it'd be great. It'd be great to see some of those in the future. So.
Awesome. Cool. With that, thank you so much, Dean, for taking the time to present an awesome seminar. We got really positive feedback in the comments. So yeah, thank you so much for coming on and being a guest. Really appreciate it.
Much appreciated.